Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and let us start our new topic, and the new topic is the electrophoresis. So, electrophoresis is a technique which is being used to analyze the biomolecules such as the protein or the DNA and to separate them based on the charge or the molecular weight or all other biochemical properties. So, let us start discussing about the electrophoresis. So, as the name suggests, the electrophoresis means the study of or the separation of the molecule when they are running into the electric field. So, you can imagine that you have a biomolecule which is, or you have a charged molecule containing Q charge and it is running into an electric field. So, electrophoresis is an electrokinetic process which separates the charged particle in a fluid using the field of the electric charge. It is most often used in life sciences to separate the protein molecules or the DNA molecule and can be achieved through the several different procedures depending on the type and size of the molecules. So, before getting into the detail of the electrophoresis and how to perform the electrophoresis, let us first discuss what is the basic principle of the electrophoresis, so that you will be able to understand each and every factor which is influencing the electrophoretic movement of or the movement of the molecule within the electric field, so that you will be able to utilize those factors to bring the better separation. So, you can imagine that a charged particle Q is moving in an electric field E, and when a charged particle is moving in an electric field, it is actually going to experience a electrophoretic force which is going to be in the direction of the electric field, and the quantity is going to be the F is equal to Q E, which is where the E is the strength of the electric field and Q is the charge of this particular particle. But when a molecule is moving in an electric field, and if the material through which this molecule is moving is also actually going to experience the frictional forces, the frictional forces are going to be oppose the movement of this molecule into the electric field, and frictional forces are going to be equivalent to the Fv, which is where the F is the frictional constant. So, F is the frictional coefficient and the V is the velocity of the electrophoretic mobility. This movement of a spherical object through a liquid media of the viscosity n, the friction of coefficient F is given by the formula 6 pi neta Rv, which means if a molecule is moving through a liquid media, which is suppose the gel, then the viscosity and the friction are going to take place, and as a result the frictional force, the frictional coefficient is going to be equivalent to, frictional forces are going to be equivalent to the 6 pi neta R and V. And now, let us imagine that this molecule will stop making the any kind of movement. So, the place where the movement of a spherical molecule of the viscosity n, the friction coefficient F is given by the 6 pi neta Rv, and the place where the particle will have both these forces equivalent, or both of these forces are equivalent, that is the place where the molecule is going to stop the movement, and that place the QE, which means the electrophoretic forces as well as the frictional forces are going to be equivalent. So, at this place the electrophoretic mobility will be given by the formula V is equivalent to Q by 6 pi neta R, whereas the Q is equivalent to the ZE, which means if the molecule is made up of the biomolecules, you can calculate the charge simply by having the valency, which means where the Z is the valency and the E is the electronic charge. So, if you multiply both the thing, you are going to get the charge, where so the electrophoretic mobility can be expressed as the ZE by the 6 pi neta Rv. So, what you see is the ZE by the 6 pi neta r, and what you see here is that this component is actually a constant, which means it is not going to be dependent onto the molecule, which means the V is directly proportional to the Z by the R. So, hence the electrophoretic mobility is directly proportional to the charge, which is this, and the inversely proportional to the viscosity of the media, size and shape of the molecule. In the case of the relative mobility, it is directly related to the charge by radius of the molecule. That is what we have said, right? V is equivalent to Z by E, which means V is directly proportional to the Z and V is inversely proportional to the 1 by R. So, this is R actually, which means and R is directly proportional to the mass of the molecule. So, for a globular protein, the radius of the molecule is directly related to the molecular mass of the macromolecule, which means the relative mobility V is directly proportional to the charge and inversely proportional to the mass. So, this is the relationship between the electrophoretic mobility and the charge of the mass can be exploited to separate the different types of molecules, because the different molecules are going to have the different charges and they also going to have the different masses. And this is the way the people have thought that if we resolve the molecules in the electric field, we can be able to separate the molecule utilizing this particular property. So, for resolving the molecules, they have designed different types of the electrophoretic apparatus, because what you are supposed to understand is that you are interested to do the electrophoresis, but you are not interested to do the electrolysis. Electrolysis is a process where the molecule will run through the electric field and then they will reach to their respective electrodes, and there the molecule is going to be broken down into the individual atoms, and that is how the molecule is going to be broken down with the help of the electric field. So, that is called as the electrolysis, whereas in the case of electrophoresis, the molecule is always remain intact and it runs through the electric field, so that it will be get separated from the other molecule based on the charge by mass. So, the first apparatus what people have designed is called as the moving boundary electrophoresis. So, in this method, the electrophoresis is carried out in solution without a supporting media. The sample is dissolved in buffer and the molecule moves to their respective counter charge electrode. Sample is loaded in the middle of the U tube and then the apparatus is connected to the external power supply. The charged molecule moves to the opposite electrode. As they pass through the refractometer, a change can be measured. As the desirable molecule passes, sample can be taken out from the apparatus along with the buffer. So, in a moving boundary electrophoresis, what they have done is they have made a U colored tube, okay, and in this U colored tube they have put the cathode on one side and the anode on the other side, and this is connected through a sample loading chamber. So, from this chamber you can actually load the samples, then the sample is going to be filled into this middle tube, and then they are going to connect the cathode and anode to the external power supply. So, what will happen is the positively charged molecules are going to run towards the cathode, whereas the negatively charged molecules are going to be moved towards the anode, and on the terminal end of the these tubings they have put the refractometers on both the sides. So, what will happen is refractometer is a apparatus which actually measures the change in the refractive index of a particular solution. So, what will happen is as soon as the molecule will pass through these refractometers, there will be change in the refractive index of this particular buffer, and as a result they will know that when a particular molecule is coming out, but that is very ambiguous, that is not very accurate, and On the other hand, there are multiple problems when they were going through with the moving boundary electrophoresis. What are these problems? The resolution of the technique is very low due to the mixing of the sample as well as the overlapping of the sample component. For example, if you load the sample in the middle, because all these is what you are doing in a buffer which is actually not having any supporting media. So, as soon as you turn on the electric field, the ions are moving towards the counter electrodes, but as soon as you turn off the power, they all start mixing together. Okay? And because it is happening in the free media, there will be a mixing of the samples, there will be no separations, and there will be no way that you know which molecule is moving or passing through the refractometer. So, because of that, it always have a problem of the ambiguity, because you have to standardize very nicely so that you will know when your molecule of interest is going through the refractometer, so that you can actually put the pipette and you could be able to take out this sample from the moving boundary electrophoresis apparatus. On the other hand, the electrophoresis technique is not good to separate and analyze the complex biological sample. Instead, it can be used to study the behavior of the molecule in a electric field, which means this apparatus was good enough to study the behavior of the molecule, which means it will say whether the molecule will move towards the cathode or molecule will move towards the anode, but it was never been the good enough to separate the complex biological mixture. For example, if you load the E. coli lysate or mammalian cells lysate, it will not be able to resolve the samples, because for resolving you do not have anything where supporting media, so that it will actually going to experience any kind of frictions or any kind of separation of the molecules. So, because of that, the moving boundary electrophoresis was not very popular, and then people have started developing the new techniques. Then they have developed the zone electrophoresis. So, in this method, an inert polymeric support media is being used between the electrode to separate and analyze the sample. The supporting media used in zone electrophoresis are absorbent paper, gel of starch, agar and polyacrylamide. The major advantage of the presence of supporting media is that it minimizes mixing of the sample and immobilization of the molecule after the electrophoresis. It makes the analysis and the purification of the molecule from the gel easier than the moving boundary electrophoresis. So, the major change what people have done when they were moving towards the zone electrophoresis is that they have started putting some kind of supporting media, which means they are putting some polymeric substances or some kind of gel, so that the molecule will immobilized onto those. So, because of that, the molecule will get separated and you can be able to visualize them, because they are immobilized onto the supporting substance, and then you can stain and de-stain those supporting substances to know what will be the pattern, and then accordingly you can optimize the separation techniques, and accordingly you can be able to achieve the better purifications. The substances what they were using for the supporting media are the paper, the starch, agar and the polyacrylamide. Accordingly, the moving boundary gel electrophoresis is also being called as the gel electrophoresis, and within the gel electrophoresis you have the two mode in which you can be able to perform the gel electrophoresis. One is called as the horizontal gel electrophoresis, which means you are performing the gel electrophoresis in the horizontal direction. So, the gel electrophoresis in this system performed in a continuous way. The classical example of this is the agarose gel electrophoresis. Then you have the vertical gel electrophoresis. So, in a vertical gel electrophoresis you are performing the electrophoresis from top to bottom. The electrophoresis in this system performed in a discontinuous way with the buffer in the upper and lower tank connected by the gel slab. It has the multiple modification in the running condition to answer the multiple analytical questions. So, This zone electrophoresis is also been called as the gel electrophoresis, and the gel electrophoresis can be performed in two different mode, either the horizontal gel electrophoresis where the system is going to be continuous, and the second is the vertical electrophoresis where you are going to have the discontinuous system where the gel is being placed between the upper and the lower tanks. So, these are the components of the vertical gel electrophoresis where you have the gel cassettes, electrode chambers, then you have the tank where you are going to place this, then you have to have the comb, then you have this the electrodes or the power cord, and then you have the power supply unit which is actually going to supply the desirable power between the electrodes. So, these are the electrodes what is being present into the electrode chamber. The reagents what you are looking for the vertical gel electrophoresis. So, the buffer and the reagent for the electrophoresis, the different buffers and reagent with their purpose for vertical gel electrophoresis is as follows. The first thing what you need is the timid or the NN tetramethylethyl and the timid is a catalyst, it catalyzes the acrylamide polymerizations. Then you require the ammonium persulfate or APS, it is an initiator of the acrylamide polymerization. Together these two components are required to polymerize the acrylamide. Then you require the tris HCl. So, tris HCl is required to prepare the running as well as the gel casting buffers. Then you have the glycine which is a component of the running buffer. Then you have the bromophenyl blue. So, bromophenyl blue is a tracking dye and it is required to monitor the progress of the gel electrophoresis. Then you require the chromacy brilliant blue R250 which is a staining dye which is actually going to stain the polyacrylamide gel. Then you require the sodium dodecyl sulphate or the SDS. It is used to denature and provide the negative charge to the proteins, and then you require the acrylamide. So, acrylamide is the monomeric unit used to prepare the gel, and then you require the bisacrylamide which is the cross linker for polymerization of the acrylamide monomer to form the gel. Now, let us see how the acrylamide is going to be polymerized. So, what you do actually is you are actually always making a gel solution which is actually the 30% acrylamide solution. So, in the 30% acrylamide solution what you do is you add the 29 grams of acrylamide and then 1 grams of the bisacrylamide, and keeping these together when you are adding the timid and APS, that actually is catalyzing the cross linking of the acrylamide monomer with the bisacrylamide. So, how the acrylamide is going to be polymerized is that you have the acrylamide and then you have the bisacrylamide, and when you are incubating these with the timid and the APS, what happen is that the APS which is ammonium persulfate in the presence of timid forms the oxygen free radicals and induces the polymerization of acrylamide monomer to form a linear polymer. These linear polymers are interconnected by the cross linking with the bisacrylamide monomer to form a 3-dimensional mesh with the pore. So, what happen is these acrylamide monomers are being polymerized. So, they will form the fibers like this. So, because these two molecules are forming the radicals, and suppose they form the radicals on one molecule, this radical is actually interacting with each other, and that is how they are actually making the polymeric fibers, and then these polymeric fibers are also being connected with the help of the bisacrylamide, because the bisacrylamide has the cross linking groups on the both side, and that is how you are actually making a 3D mesh which is actually containing the pores in between, and these pores are always being used for the biomolecule to the pass through. So, because of this mesh you can be able to experience or you can be able to produce the friction for the different types of molecule, and that is how the friction is going to play in separation of the molecule. So, because this mesh has the sizes of different sizes, these mesh is going to play a kind of a separation filter or something like that. So, that it will actually going to help in getting the separation of the biomolecules. The size of the pore is controlled by the concentration of the acrylamide and the amount of bisacrylamide, which means if you increase the bisacrylamide concentration or if you increase the acrylamide concentration, you are actually going to decrease the pores, because you will be keep putting more and more fiber, and as a result the pore size is going to be smaller and smaller, which means you cannot be able to use a very highly cross linked gel with a small with a large protein, because if you use the large protein will not be able to enter into these pores, and as a result it will be get excluded from the gel. In a vertical gel electrophoresis system, we cast two different types of gel, stacking gel and the resolving gel. First, the resolving gel solution is prepared and poured into the gel cassette for polymerization. A thin layer of organic solvents such as butanol or the isopropanol is layered on the top to stop the entry of oxygen. Why it is so? Because the oxygen neutralizes the free radicals and slow down the polymerization and makes the top layer smooth. After polymerization of the resolving gel, a stacking gel is poured and the comb is fitted into gel for construction of the different lanes for the sample. So, the running of a vertical gel electrophoresis, as we I think discussed, you need a gel cassette, then you need an electrode chamber, then you need a tank, and what you do actually is that you first pour the resolving gel, because these are the gel which is going to use for separation of the molecule, and then on top of that you are actually going to put the stacking gel. The difference between the resolving and the stacking gel is in terms of the composition of the acrylamide as well as the pH of the buffer what you use for these two gels are also different. And then when you cast the resolving gel, you actually put the a layer of the organic solvent, so that it actually helps in terms of the polymerization of the acrylamide. The running of the gel. The sample is prepared in the loading die, which contains the SDS, which means the denaturating agents, beta mercaptoethanol, which is actually going to break the disulfide linkages, and you need, and all these will be present in the glycerol, because the glycerol is required to provide the density in the sample, so that it facilitate the loading of the sample in the well. Now, since these wells, so you can imagine that this is a typical well where you have loaded the different types of samples. Since all the samples are filled vertically, there is a distance drift between the molecule at the top versus the molecule at the bottom inner lane. So, you can imagine that you have, this is a lane where you have different types of molecules, and all these molecules are having, for example, a molecule number 3 and the molecule number R. All these molecules, imagine if the 3 and R of a same molecular weight, even then there will be a drift of the distances between the two, because so if you resolve them without going through the stacking of the sample or without putting them at the same place for running, they will be having a drift. You can understand that simply by looking at the racing track. So, if you see a racing track, what you see is that the players are sitting at a very different locations, because all these different locations the diameter of the circle is different, but in this case you do not have such options. So, what you have to do is you by any mean you have to bring the 3 and the R together, and that you can do simply if you do a stacking of the sample. Now, question is how you can be able to stack the sample. So, to stacking of the sample you have to first put a stacking gel. So, that is why the well has to be prepared in the stacking gel, and you have to stack the sample. And why the stacking gel is actually doing the stacking? Because the pH of the stacking gel is 6.8, and at this pH the glycine is moving slowly in the front, whereas the tris HCl is moving very fast. So, you can imagine that you have this well where this is the sample number 3 and this is the sample number R, which is very far away, but these samples are present in a buffer which is actually having a composition of tris HCl and glycine. So, and the pH of this gel is 6.8, which is actually the pH of the stacking gel. So, at 6.8 the glycine is having the very low electrophoretic mobility. Because of that, there will be a blockage of glycine in the front. So, you have a glycine which molecule which are sitting in the front, and then you have the tris molecule which are actually present in the from the top. So, because of that, you can imagine that you are putting the molecules, the two molecules like the number 3 molecule and the R molecule between the, like where the tris is going to be used as a plunger. So, what happen is the tris is pushing the molecules, whereas the glycine is not allowing these molecules to move as per their electrophoretic mobility. So, because of that, all the molecule will reach to this point and they will be able to get stacked. But what happen is, and this will happen because they will be keep running into the stacking gel for some time, and during this period only the 3 and R are going to come together and they will form a single band, which is actually going to contain the 3 and R together, and that will happen while they are running into the stacking gel. But as soon as they will enter into the resolving gel, so after this they will enter into the resolving gel. So, resolving gel has a pH of 8.8, which means at this pH the glycine is now charged and it moves fast, and now sample run as per their molecular weight, because the SDS is going to provide them the equal negative charge. After the tracking dye reaches to the bottom of the gel, gel is taken out from the glass plate with the help of a spatula, and it is stained with the Comasy Brilliant Blue R250, the dye stains protein present on the gel. So, as soon as it enters into the stacking dye, this blockage of the glycine is going to be removed, and as a result the proteins are, the molecules are going to be resolved based on the molecular weight. The higher molecular weight are going to run slower and the smaller molecular weight are going to run smaller. And because you want to know how much the molecules are going to traveling into the resolving gel, you are also using the tracking dye, that is the bromophenol blue. And so, bromophenol blue is a very small dye. So, it runs in the front, and as soon as it reaches to the end of the gel, you will be able to know that okay, the electrophoresis is now over, and then I can stop the gel and I can take out the gel, and I can do the staining and destaining and to see what is the pattern of the gel. So, this is all about the theoretical information of the electrophoresis, how the history of the electrophoretic apparatus, how the apparatus is being evolved by exploiting the or by developing the different types of electrophoretic apparatus. So, but this is all the theoretical knowledge. Now, I would like to take you to my lab and I would like to show you a small demo how to cast the gels and how to perform the electrophoresis. And in this demo, the students have resolved the samples, and then they have also shown you how to stain and destain.
In this video, we will demonstrate you how to run a SDS page gel, and how to prepare various reagents required for the running of SDS page gel, and what are the different instruments we can use. So here, this is the gel casting stand. So where we can use these glass plates to prepare the gel. In between there is a space where we can pour our gel solution. Then we will keep for some time, at least 20 to 30 minutes, let it solidify. Then we will prepare stacking gel. Then we will load the our protein solution. So here, before doing that, we need some reagents. So what are those reagents? The first reagent we need for this experiment is acrylamide. So generally we will prepare acrylamide 30%. 30% means 29 grams of acrylamide and 1 gram of bisacrylamide. These both we can use 29:1 ratio in 100 ml of water to get 30% of acrylamide. So both these are neurotoxic, so we have to wear gloves always. After this, we have to prepare resolving gel. For resolving gel, we need 1.5 molar Tris HCl, pH 8.8. In addition to that, we also need 10% SDS prepared in double distilled water, and also 10% ammonium persulfate and also timid. The role of ammonium persulfate and timid we can see during preparation of gel. They act as a catalyst. After solidifying, we have to prepare stacking gel. So stacking gel is nothing but composition is same, but we can say it is diluted. It contains pH 6.8 Tris HCl and remaining components same but in less quantities. So after preparing the gel, we will load the marker and the protein, which is denatured at 100 degrees Celsius for 3 minutes. After that, we will fix this gel into this one. We will keep in this reservoir. Then we will connect to the power pack and run the gel. So this is the overall introduction of how to prepare a SDS based gel. So let's start the preparing gel. We will learn more things while preparing the gel. Before preparing the resolving gel, we have to prepare the casting, set up the casting gel. So this is the glass plates. This is very thin one. So this is the main glass plate. This is 1.5 mm glass plate. It is available in 1 mm glass plates also. If your loading solution is less, like you want to load only 20 microliter, 30 microliter, then 1 mm gel is good enough. But if you have extended volumes like 70 microliter, you can use 1.5 mm gel. You have to arrange like this short plates on this plate, and the bottoms should be equal. Then we have to put in this one, this tray. Then we are going to keep like this. So we have to check. If we perfectly set up this one, then there should not be any leakage. But if there is any leakage, your resolving gel may leak out and you will get nothing. So in that case, we have to check it prior to pouring the gel, so whether it is okay or not. So I am going to use milky water. So just after checking the gel, is there any leakages or not. So we moved forward for preparing a resolving gel. So the composition is given in this slide. Please go through that slide. This is just water. First I used water. I am going to add sequentially 4 ml of water. Now I have to add 3.3 ml of already prepared 30% acrylamide. Already in introduction, I explained how much percentage we have to prepare and how much quantities of acrylamide and bisacrylamide need to take. So here we have to add 3.3 ml of acrylamide solution, 30%. So I have to adjust 300 microliter. The next component is 1.5 molar tris pH 8.8. We have to add 2.5 ml. Next component is SDS. Here SDS functions as plays as dual role. Like one thing is that it gives negative charge, gross negative charge on the polypeptide chain. The next component we have to add is SDS, 10% SDS. We have to add 100 microliter of SDS to resolving gel. It plays very crucial role in polyacrylamide gel electrophoresis. Like it imparts negative charge on the polypeptide chain, so that despite of their charge, they will move based on the molecular weight. So I am going to add SDS. The other important thing is that 10% ammonium persulfate. Ammonium persulfate, which is catalyzed by the timid, provides free radical species which accelerate the forming mesh-like shape in acrylamide gel. Like it will catalyze forming the mesh. So this is the 10% APS. I just add 100 microliter of 10% APS to resolving gel. In final step, we have to add timid. Timid, after adding all the components, at the end of the gel, we have to add timid, because if you add earlier, it will quickly facilitate the polymerization. So you cannot take out with the pipette. So it completely solidifies. So that's why you have to add at the end of the resolving. So I'm going to add 5 microliter of this timid, which catalyzes the ammonium persulfate. Ammonium persulfate in turn provides free radical species, and free radical species accelerate the polymerization. This is the overall principle of this resolving gel. So I will add timid. We have to mix properly. Then add slowly at one corner. So after this we have to overlay with, on the top layer we have to overlay with some solvent like 2-butanol or isopropanol or with water. So why we are doing this? Because if the gel is exposed to air, then the oxygen from the air will interfere in the polymerization of the gel. So we have to add either water or 2-butanol for this purpose. Now we have to check whether it is solidified or not. So it is solidified. Now we have to remove the overlay layer, like we have used water, so no need to remove. If you are using isopropanol or butanol, you have to remove that and wash with the milky water. So now we will start preparing the stacking gel. The compositions are given in the video. You have to add 3.4 ml of water first. Next 830 microliter of acrylamide. Six 30 microliter of Tris HCl pH 6.8. Fifty microliter of timid and 50 microliter of SDS we have to add. At the end we have to add 5 microliter of timid. We have to mix properly after adding the timid. Then you just add at one corner. Next we will keep calm. Now we will wait until the gel got solidified, then we will shift to the buffer tank and then we will run the gel. While the stacking gel is solidifying, we have to prepare sample for loading in SDS plain gel. So for that we have to prepare loading die, 10x or 6x loading die. It mainly contains 250 millimolar tris, pH 6.8, 30% glycerol, 10% SDS and 0.05% of bromophenol blue. So here we can add 10 millimole of DTT also as a reducing agent. SDS mainly works as imparting negative charge on the polypeptide chain, and DTT breaks down the disulfide bonds. If you have a dimer which you can see as a monomer in SDS page. Suppose you have 20 kDa, that means 40 kDa protein, which is a dimer actually. You can see only 20 kDa band corresponding to that protein, because DTT breaks down the disulfide bond and you can see only single band. If you want to see actual molecular weight, you have to run it on native phase where there is no reducing agent or no detergent. The other thing is glycerol. While loading the gel, since the protein solution is not that much dense, it may come out from well. So in order to prevent this thing, we have to load with the denser solution like glycerol. So 30 to 50% glycerol is sufficient for keeping the protein solution intact in the bottom of the well. So other thing, bromophenol blue. Bromophenol blue we use for just tracking the how much gel completed. So this is the loading die. So we have to take the protein solution. Here we already prepared 10% of loading die. So that means this is 10x loading die. We have to prepare 1x to mix with the protein solution. So this is 100 ml of solution, which loading solution. We mix 10 microliter of loading die to this protein solution. You can tap down or pipette this protein solution. Then we have to heat it for 3 minutes at 100 degrees Celsius, so that all the polypeptide chains, dimers are, if any multimers are present, they will break down and we can see nice band. So I am going to heat this at 100 degrees Celsius for 3 minutes. This is the remaining of stacking gel solution. So we can see it is solidified. So that means the stacking gel also got solidified. We have to remove that gel and fix it into this one, and we have to keep inside the tank. So I just take out the gel. So inside this tank we only have this side one. You have to cover other side also. So for that we use a dummy plate. You just hold it tight and close this thing. After that, gradually adjust the gel length. So just we have to fix like this. Once fixing here, we have to add this running buffer. The running buffer contains 15 grams of trace, 72 grams of glycine and 5 grams of SDS for 2 liters of solution. 1x solution. So this is 1x I already prepared. I am going to add. We added in this tank, but the main tank surrounding to this one, we have to add up to the mark. So for reference, you can see here, for 4 gels we have to add till here the buffer, we have to load outside this gel. So for 2 gels here, for 1 gel we can add like this. This is the power pack where we can adjust the how many volts we want to run. The protein samples are ready. We heated sufficient time. Now we have to load this. So we have to remove the comb carefully. Then first I am going to load marker or protein ladder. Next I will load SAM. Once the loading goes over, we have to fix this gasket. I am going to set it at 70 volts. As we can see, it is almost over. So we can take out the gel, then we will stain and de-stain it. Generally what we will do is, we will there are two ways of staining and de-staining process. One is we can do quick staining, like we have to heat it with the staining solution which contains Kumasi Brilliant Blue and along with methanol and water. So then we will try to de-stain with the water by heating. But in another way, the simplest way is, we will just stain the gel for 2 hours, then we will de-stain overnight. So I am going to show the simplest way. First we will stain in Kumasi Brilliant Blue staining solution, then we will de-stain in methanol water containing salt. So I am going to stop the children, then I will remove it. I will show you how to remove the gel. Take out the glass plates. Here we have to be very careful while taking out gel, otherwise the short plates may break. On a corner we have to take and lift the gel like this. So keep the gel in a staining box, which is more or less a plastic one, but it can sustain the. So then I am going to add staining solution. I will keep it for a rotation on a shaker for at least 2 hours, then we will de-stain over. So once the time is over, after 2 hours, we will de-stain this solution. Now we kept 2 hours in staining solution. As we can see, the staining is over. Like you can see, the gel completely turned into blue. So we remove the solution. Then I am going to add de-staining solution. And I will keep this on a rocker for 2 hours for de-staining. So the composition contains for 100 ml of de-staining solution, 40 ml of water, double distilled water, and 40 ml of methanol, and 10 ml of glacial acetic acid. So I am going to keep this on a rocker. We have run the gel and stained and de-stained. Now we will capture the gel image. So we can see manually also, but for record purpose we have to capture it through GelDAC. So this is the GelDAC imaging system from Biorad. So I'll show you how to capture the images. So here we will use white tray. There is another one gray or UV tray is also there. So there you can see any fluorescent one or stained with the ethidium bromide or blots, chemiluminescent blots, you can use that. But for normal protein imaging, we can use this white tray. So I am going to keep the gel on this one. So we have to open properly. This is very important step. You have to align the tray in a proper way. So otherwise it will show error. So once it is over, you just push it back. So we have to log on to account. So this is SDS page gel. You can select the application whatever you want. So here nucleic acids, protein gels, blots, 3 different categories are there. So we are observing here protein gels. Protein gels stained with the Kumasi blue or white tray. We are using white tray, so this is the right tray. You can use Kumasi blue stained one gray tray also, but we are using white tray, so we will use Kumasi blue. So auto optimal, then I will ask for capture. So it will take 1 to 3 minutes based on the signal intensity. So as we can see, it is optimizing the signal intensity. You can minimize this one also so that you can see the gel image. So now it is over. If you want to do any modifications to images, for suppose you want to decrease or increase the signal intensity, so this kind of changes you can do. So if you want to send this gel, you can have send and save. If you have any drive connected to this one, you can send directly to this one, that thing. So for image analysis part, we'll show in the upcoming video how to analyze what this band of interest correspond to which molecular weight. So we already loaded the molecular weight one, so we can easily find out using ImageLab software. In this video, we have learned that how to prepare a SDS page gel and how to run it. What are the precautions need to be taken while preparing the gel, and how to observe, how to record the gel using gel documentation system. So I hope this will give you a gist of how to prepare and run a SDS page gel and analyze the protein sample.
So with this demo, we have shown you the different steps what is required to prepare the acrylamide solutions, and then how to prepare the resolving gel and the stacking gel, how to load the samples, and then how to perform the electrophoresis, and then ultimately we have also shown how to do the staining and destaining. I hope that this demo video could be helpful to understand the electrophoresis, the vertical gel electrophoresis. So with this, we would like to conclude our lecture here. In our subsequent lecture, we are going to discuss about the imaging and the staining and destaining, and how to analyze the gel pictures what you are going to capture just after the electrophoresis. So, with this I would like to conclude our lecture here. Thank you.